Have you ever thought bacteria also have a preferable temperature?
Like us preferring air conditioning instead of hot weather?
Actually, yes. Temperature really affects how fast they grow.
And that's what our project is about, tracking microbial growth.
We are from Group 1.
I'm Stephanie.
I'm Lin Zhi'en.
I'm Gong Mingling.
I'm Shafiq.
Let's begin!
Microorganisms, although invisible to the naked eyes, are constantly growing and reproducing around us. This process is known as microbial growth, where microorganisms such as bacteria increase in number through cell division. Among the many factors that influence their growth, temperature plays a very important role. Just as humans feel more comfortable in certain environments, microorganisms also have their own preferred temperature range. When the temperature is optimal, they grow rapidly. When it is too low, their growth slows down, and when it is too high, they may die. The purpose of this assignment is to investigate how temperature affects microbial growth and to demonstrate its importance through our video project, Tracking Microbial Growth.
Before starting the experiment, we ensure proper safety and aseptic preparation by washing and drying our hands thoroughly. We then wore appropriate personal protective equipment such as lab coats, gloves, face masks, and cover shoes. All hair was tied up properly to avoid interference during the experiment. To maintain aseptic condition, the working bench and glove hands were disinfected using 70% alcohol before handling any materials. To limit neutron broth and sterile peptide tips, the neutron broth was first transferred into sterile centrifuge tubes for easier handling. During the transfer process, aseptic techniques were strictly followed by flaming the openness of the neutron broth bottle and sterilize test tube with a Bunsen burner to minimize contamination and maintain sterility throughout the experiment.
To carry out this experiment, we prepared several material and equipment to observe microbial growth under controlled conditions. This experiment used Escherichia coli culture nutrient broth. The apparatus prepared include sterile 10 ml measuring cylinders, micropipette, 100 microliters, and 1 ml with sterile tips, cuvettes, sterile test tube, and test tube rack. The equipment used were a 37 Celsius incubator, spectrophotometer, Bunsen burner, and vortex mixer. First, we prepared 3 sterile test tubes, 10 ml each for each condition, and label them according to the experimental setup for Escherichia coli. We inoculate each test tube by transferring 0.1 ml of bacterial suspension using a sterile pipette. For the control samples, we prepare a positive control consisting of nutrient broth, inoculate with bacteria, and incubate at 37 Celsius as this temperature was expected to support optimal microbial growth. We also prepare a negative control containing only sterile nutrient broth, which serves as the blank for turbidity and body measurements. All test tubes were incubate at different temperatures, including 4 Celsius, 37 Celsius, 60 Celsius, and negative control for 24 and 48 hours to observe the effect of temperature on microbial growth. After inoculation, each sample was mixed thoroughly using a vortex at medium speed. We transferred the sample from different temperature conditions into separate cuvettes for observation after 24 hours. We then observe and compare the turbidity of each sample against the inoculate broth. Finally, we measure the optical density, or D600, of each sample using spectrophotometer set at 600 nanometer using the inoculate nutrient broth as the blank control.
After 24 hours of incubation, the test tubes from different temperatures were carefully removed and placed into a test tube rack for observation. To record the results, we used a growth scale table to estimate and classify the turbidity of each sample based on visual comparison. Next, we prepared 12 cuvettes for optical density measurements using the spectrophotometer. Before transferring the samples, each cuvette was rinsed with nutrient broth. This step helps to remove any residual contaminants or water droplets and ensure that the sample being measured is not diluted or affected by foreign substances. Then, using a micropipette, we transferred 3 ml of each sample into individual cuvettes. Before placing the cuvettes into the spectrophotometer, the transparent sides were wiped using Kimtech tissue paper to remove fingerprints that may interfere with light transmission and affect the readings. We then placed one control cuvette and 3 sample cuvettes from the same temperature group into the spectrophotometer. The machine was set to 0 first. Then, we proceeded with the measurements by pulling the level 3 times and recording the absorbance values. All data was saved and later viewed for analysis. After measuring the sample, the test tubes were re-incubated for another 24 hours. After 48 hours, the same procedure was repeated to observe the turbidity of each test tube and recorded OD600 readings. However, one of the 37 degrees Celsius samples showed an OD600 value greater than 1, which is beyond the reliable measurements range of the spectrophotometer. To solve this, we performed a dilution using distilled water. We added 1.5 ml sample into 1.5 ml of distilled water, making a total of 3 ml into the cuvettes to reduce the concentration of the sample. After After dilution, the sample was measured again using the same procedure, and the OD600 value was recorded accordingly. Based on our observations, most samples show microbial growth patterns that match our expectations. However, one negative control sample showed unexpected turbidity after 24 hours, indicating possible contamination during handling. After another 24 hours of incubation, the turbidity became more obvious.
Before comparing the result, it's important to understand the relationship between turbidity and adsorbent. When the solution is more turbid, it means there are more bacterial cells present. As a result, more light is absorbed and less light passed through with a higher adsorbent reading. On the other hand, when the solution is less turbid, fewer bacterial cells are present, so more light can pass through, resulting in a lower adsorbent value. Therefore, the spectrophotometer provides a quantitative and more reliable measurement of microbial growth compared to visual observations alone, as it directly reflects cell density in the culture. At 37 degrees Celsius, which is our positive control, the bacteria saw the highest growth at both 24 and 48 hours. The average adsorbent value were 0.797 plus minus 0.106 and 0.771 plus minus 0.181 respectively. This indicates high turbidity and a large number of bacterial cells, which can be classified as nutritious growth. In contrast, at 4 degrees Celsius, the adsorbent value were very low, with average of 0.0413 plus minus 0.019 at 24 hours and 0.032 plus minus 0.020 at 48 hours. This showed that bacterial growth was significantly slowed down due to reduced enzyme activity at low temperature. At 60 degrees Celsius, the adsorbent value remained very low, with average of 0.018 plus minus 0.009 at 24 hours and 0.019 plus minus 0.004 at 84 hours. This suggests that the high temperatures inhibited or even killed the bacteria, preventing growth. For the negative control, which should not show any growth, turbidity was observed at 24 hours. This was unexpected and suggests possible contamination during the experiment. After 48 hours, the turbidity became more obvious, suggesting that the contamination increased over time. Any deviation such as the contamination observed in the negative control highlight the importance of strict aseptic technique in ensuring experimental accuracy. To prevent contamination, precautions such as sterilizing equipment properly, minimizing exposure of culture media to the environment, and handling sample carefully during transfer should be strictly followed throughout the experiment. This result showed that temperature is a key environmental factor affecting microbial growth. Each microorganism has an optimum temperature range where it grows best. Escherichia coli is classified as a mesophilic bacterium, meaning it grows best at moderate temperature, typically between 15 degrees Celsius and 50 degrees Celsius. This explains why the highest growth was observed under this condition, as it reflects in natural habitat in the human intestine. In real-world application, this principle is very important. For example, refrigerations at low temperatures slow down microbial growth and help preserve food, while high temperatures used in cooking can kill microorganisms and improve food safety. In food safety, storing food at temperature close to 37 degrees Celsius can promote rapid bacterial growth and increase the risk of foodborne illness. In clinical setting, understanding the optimal growth conditions of bacteria is important for culturing and identifying microorganisms. Overall, our results are consistent with previous studies, which show that bacteria grow best at moderate temperatures and are inhibited at extreme temperatures. This result also demonstrates that controlling temperature conditions is essential for regulating microbial growth, which is a key principle in microbiology, food safety, and clinical application.
After completing the experiment, all microbial waste was disposed of properly in a designated biohazard container. The working bench and equipment were cleaned and disinfected, and all apparatus were returned to their original places.
In conclusion, our experiment showed that the temperature has a significant effect on microbial growth. The results clearly indicate that E. coli grow most effectively at 37 degrees Celsius, which was its optimal temperature, while minimal or no growth was observed at 4 degrees Celsius, 60 degrees Celsius, and in the negative control. However, there were several limitations during the experiment. One limitation was the limit amount of mutant growth and pipette tips, which require careful handling of materials throughout the experiment. In addition, human error during pipetting and handling may have affected the accuracy of OD600 readings. To improve this experiment, we recommend using more precise automated pipettes and increasing the number of replicates to ensure more reliable results. From this study, we learned the importance of temperature control in microbial growth and how temperature significantly influences bacterial activity. We also gained hands-on experience in aseptic techniques, laboratory safety, and the use of instruments such as the spectrophotometer.
Overall, this experiment helped us better understand how scientific methods are applied to study microbial growth.